Hi, hello, Year 9, and welcome to another science lesson. We are on week 4, and this is lesson number 4, the last of the PowerPoints for the week, because tomorrow Miss Corbett will be doing a video showing you the required practical to do with enzyme activity. Okay, so what we're going to be looking at directly relates to that practical, because we're going to be looking at factors that affect enzymes, so things that will have an impact on how quickly those enzymes work or the rate of reaction. Now, before we begin, what I would like you to do is match up all of the keywords with the definitions. These are all words that we came across in last lesson. You do not need to write them all out. As you can see, I've put letters on the keyword and numbers on the definition. So if you pause the video and just write down the letters and then which number it correlates to. Okay, so hopefully we've had a go at that, just trying to remember some of the things we looked at yesterday. And here are the answers. So if you grab a different colour pen, you can see whether you've got them correct. I would suggest that if there are any that you did not get correct and you do not have in your notes from last lesson, that you just pause the video and just write down that keyword with its definition. Okay, so here we go. The answers are on the right hand side. So A for active site, that is number 7, the part of the molecule where the reaction occurs. B, which the keyword was denature, goes with the number 6, which is active site of the enzyme changes shape. It can no longer act on the substrate. C is enzyme, and that goes with number 5, which is proteins found in living organisms that speed up the rate of a chemical reaction. D is temperature, and that goes with number 1, which is how hot or cold something is. E for pH is number 2, the level of how acidic or alkaline a substance is. F, the lock and key mechanism or the lock and key theory, goes with number 4, where the substrate fits into an active site. G is specific, and that goes with number 3, relating to a particular type. I for substrate, goes with number 9, molecule on which an enzyme acts. And J, catalyst, goes with number 8, a substance added to a chemical reaction to alter the reaction rate without being used up in the process. So hopefully we got all of those correct. They've all been referred to in previous lessons. As I say, if there's any that you didn't get right, just pause the video and make sure you've got that keyword and definition in your notes. Okay, so we're going to be looking at 2 factors mostly. We did speak about temperature in the last lesson, but we need to know about 2 factors that affect enzyme activity. So we need to know that temperature and pH affects the enzyme activity. You're going to be doing about the method and the practical in next lesson. We need to explain why extremes of temperature and pHs causes enzymes to not work. And then for 7 to 8, which is more to do, as I say, with next lesson, is looking at an experiment we can do to investigate the effect of different conditions on enzymes. Okay, so something that will be new to some of you, but was some of the things that were covered in the chemistry topic. What factors could affect the rate of a reaction? So think back to chemistry where you're doing rates of reaction. Can you think of any factors or the different factors that will affect the rate of a reaction? Okay, so you might have said about the concentration or the pressure if it's in a gas. You might have talked about the temperature, so if there's an increase in temperature, the particles are moving around faster, so they're more likely to collide. You might have spoken about surface area, so the bigger the surface area, the more particles there are that are exposed, so they're more likely to collide and have successful collisions. Something that you wouldn't have done though in the chemistry one, but is a factor that affects the rate of reaction and is very important when we're looking at enzymes, is the pH. Okay. Now, because different organisms live in different areas, and because we have lots of different conditions in terms of pH inside our bodies, means that different enzymes tend to work better in different conditions, and that's how we've evolved to produce these enzymes that are adapted for the conditions that we are living in. Now, you don't need to write any of this down. Something that comes up in a later topic of biology is the term called extremophiles. Now, extremophiles are organisms that live in extreme conditions. Okay, so most of these are prokaryotes. So as we looked at in one of the first lessons, those are organisms that do not have a nucleus, so things like the bacteria. Quite often they live in very extreme conditions. So ones with really, really high levels of salt or extremes of temperature. So they live in incredibly cold conditions in the poles, but also in really hot conditions, such as in natural springs and things like that. They have adapted to produce these enzymes that work in these different conditions. And we can use that as scientists for our own benefit. So there's something called a polymerase chain reaction. Again, you do not need to write this down or know this, but it's just as an example, which is a really important process when people are doing DNA fingerprinting, okay, or if we're looking at the Human Genome Project. And they actually use enzymes from bacteria that live in these extreme conditions. And these are the ones that live in really hot conditions. So when they are carrying out PCR in really, really high temperatures, it means they can use these enzymes to help the reaction. Whereas if they were using enzymes from us, they wouldn't be able to do it because they would denature, the active site would change shape, so it wouldn't work, and obviously would be a bit useless. Now, we spoke more about the temperature in the last lesson and how temperature affects it, but another factor that affects enzyme activity that we need to be able to talk about is pH. Okay, now this is a different shaped graph that you can see. With the temperature one, it started off very similar. Would gradually get higher and higher and higher, but then would drop. It wasn't a symmetrical graph, but you can see on the pH one is very symmetrical. So it just goes up, reaches the optimum pH, and then falls rapidly. Okay, so this is what the graph would look like for how pH affects enzyme activity. Now you can see that this particular enzyme that's being used has an optimum pH of 8. So if you were asked that in an exam, you need to find the peak of the curve and you need to go down from there. You can see that on the dotted line, and you need to read off whatever pH is at the bottom. And different enzymes work in different, or have optimums at different pHs. Now, the same as with temperature. If the pH, but with temperature, sorry, it's the enzymes are denatured. If it's too high, they just work really slowly if it's too low. But if you get these extreme extremes in the pH, again, it causes the enzymes to change shape, and it's the active site where the substrate binds to it where the shape change is so important. So the active site changes shape. That means the substrate will no longer fit into the enzyme and therefore will not be working. And remember, we call that denaturing. The enzyme has been denatured. Now, I would suggest getting down a little sketch of that graph for how pH affects the rate of reaction on enzymes, and just put a little annotation about what is happening where the graph is going down. Okay, so that is where the shape of the active site is changing and the substrate can't fit into it. It has been denatured. You might want to pause the video to do that. Okay. All of the enzymes, as I said, will have a different optimum pH, and most of them will be around pH 7, which, as you know, is neutral. But there are certain enzymes that prefer higher pHs or very low acidic pHs. So one of the group, I suppose, the ones that we produce in our stomach, because we have stomach acid, which brings the pH to about pH 2, they work really well in low pHs, and one of those is called pepsin. Okay, so just a quick recap, putting it together. We've got a graph that is showing the effects of pH on the enzyme activity. So you don't need to draw this graph, it's exactly the same as the one you have done, but without looking back at the notes that you've just made, for 3 marks, can you describe and explain the effects of pH on enzyme activity? So just put it all together as though you're answering an exam question, with the exam question being describe and explain the effects of pH on enzyme activity. And we need to get 3 points in there. So pause the video and have a go at that. Okay. So on the previous one, hopefully you have got about as the pH increases, the enzyme activity increases, reaching the optimum pH of 8. And then in the extremes of pH, so if it's too low from the optimum or too high from the optimum, we've spoken about the denaturing of the enzyme where the active site has changed shape. Okay, another question for you. These are taken from exams. This one is referring to temperature, which we looked at in last lesson. And I've pointed out 3 really important parts of the graph. The bit going up, the bit at the top, the B, and then the bit going down for C. So what I would like you to do, without, again, looking back at the notes from last lesson, can you describe what is happening at each stage and also explain why the rate of reaction changes? So what is happening and why it is happening at each stage from what you remember. So pause the video now and have a go at that. Okay, so grab a different colour pen and we'll go through and see how we did in terms of how temperature affects the rate of enzyme activity. Now, I've kind of put this together. So we've got what's happening at A with a describe and explain, what's happening at B with a describe and explain, and then again for C. So at A, at the initial part of the graph, in your description, you should be talking about how the rate of reaction is increasing as temperature increases. Now, it's really important when you have these graph questions in exams that we're looking at the axis. So try and get your sentence to do with as whatever is on the x-axis increases, what happens to the y-axis. Now, the increase in temperature means that there's more energy. Okay, and you might say kinetic energy, the movement energy, but if you're talking about energy, that's great. And it means that the substrate and the enzyme molecules are more likely to come together, collide, which means the rate of reaction goes up. So that would be a lovely answer for describing and explaining that upward trend on the graph. Now the next bit is looking at the optimum, and just saying that that is the optimum temperature will get you marks in the exam. So do make sure that at the peak you are talking about that being the optimum temperature. Now you should also make sure that you put in the temperature for the optimum, or pH if it's talking about pH. So from the graph we can see that 40 degrees Celsius is the optimum temperature for it, and that's where the rate of reaction is the fastest, or it's the maximum rate of reaction. And then finally, talking about the line on the graph going down, we could talk about after 40 degrees Celsius, something about the active site changing shape. That causes the enzyme to be denatured, or denaturation. And that's where the substrate is no longer fitting into the active site. And at 60 degrees Celsius, there is no reaction happening at all, because all of the enzymes have been denatured. So hopefully we're able to describe what is happening. So that is just where you're looking at the graph, talking about what the shape of that graph means, and then also explaining, saying why that is happening. Very, very different command words, describe and explain. So do make sure that you are practicing the right answers for them. Okay, so this one is slightly different, this graph. We have got a graph showing the rate of reaction at different pHs, but we've actually got two different enzymes on here. So pepsin is a good one to remember for the low pHs, and that is shown by the orange curve going up and then down. And then we've got the enzyme amylase, which is the blue line, and that is obviously at a higher pH. So very, very common question again is comparing different enzymes. So here you've got the two enzymes, and the question is, how does the effect of pH differ for pepsin and amylase? Now I suggest that when you're answering this, you also talk about where you might find these different enzymes. So we've spoken about both these enzymes before. So think about where they're produced and why they would have that optimum. Okay, so we're going to go over the answer now. I didn't say pause the video, so if you are still playing this, pause the video and then write your answer down for this, and then we'll have a look at what it means. So grab that other coloured pen or pencil and we'll go through. Okay, so the main things to take from that is that the optimum pH of amylase is a pH of 7. Sorry, that should not be a capital P there. The amylase optimum is pH 7, so it works better at higher pH levels. And we actually find amylase, it's the enzyme that we produce in our mouth. So the pH of our mouth is neutral. We also produce it in our small intestines. And again, that has more of a neutral to alkaline pH. The other one, pepsin, works at a lower pH. Its optimum was pH 2. And these enzymes function well in the stomach, where you have hydrochloric acid. So hopefully in your answers, you have spoken about that. You've picked out the optimum pHs for both of them, so 7 and 2, and that we've spoken about where you might find them in the body. Okay, so as there's been quite a few long answers on there, this is going to be the last task for today's lesson. So I've got 5 sentence starters here. I would like you to finish the sentences as best you can, and then we'll obviously go over the answers afterwards. So if you could pause the video now, and if you could have a go at completing those sentences with using as much scientific terminology as we can. Pause the video now. Okay, so hopefully we've had a go at those 5 sentences, and we have got something that looks like this. So for question 1, it was increasing the temperature gives particles more energy. Okay, now you might have specifically said kinetic energy. That will be fantastic. That's the movement energy, but it does give the particles more energy. Number 2, high temperatures enzymes denature. Now you might have said that they work faster, or it's got a faster rate of reaction, but it's this question is mainly talking about the extremes of temperature. So if you put faster rate of reaction, you're talking about how increased temperature makes it faster. Then I can understand that, but it's talking about the extreme temperature. Number 3, the optimum temperature is, and that's when the reaction goes at its fastest rate. Okay, so when the reaction is going fastest. Number 4, there is a range on this. You don't have to specifically have said 37. Amylase works best if you put anything between sort of 37, 37.5, or even 40. I'll accept that because that's about body temperature. Number 5, if an enzyme is denatured, the active site changes shape. That's such an important sentence. Make sure that you talk about the active site changing shape. Anything to do with denaturing, the active site changes shape. So it doesn't function anymore, and it's irreversible. So we haven't really spoken about that because there will be obvious on that, but it means you can't go back again. So once the enzyme has been denatured, that's it for the enzyme. It will not be able to go back to its original shape, so it will not work. Okay, so hopefully we've now taken on board, taken a little bit more about the temperature and how that affects the rate of reaction, but also know what a graph would look like for the pH, which is slightly different to the temperature one, and how to use that graph to read the optimum pHs. Hopefully we can give an example of one that works at really low pHs, so pepsin in the stomach. It's a really good one to remember, and also how different enzymes will have different optimum pHs. Okay, so next lesson, for lesson number 5, Miss Corbett will be going over the required practical for this. Now I have this time put the dodder work to be issued on Friday, so you will see that on Friday, along with Miss Corbett's lesson, video lesson, the dodder work will also appear, and the reason for that is that quite a few people were doing the dodder work before all of the lessons were published. So it's best to do after you've gone through these lessons. So I'm going to be issuing them on a Friday because some people might be ill at the time. I've given 10 days for you to complete that work, and then I will record that on the Show My Homework. Okay, so that is it for me for this week. You'll be tuning in next week for week 5, where we'll be moving on with our biology. Have a lovely weekend. I hope you're all well. Take care of yourselves and your family. Goodbye.